Okay, so now use this microphone a little bit. So I used the term, I said that some T cell receptors are class 1 restricted and some are class 2 restricted. And we explained that when we say that the CD8 T cells, CD8 T cells, we say that their T cell receptor is MHC1 restricted. That was a great puzzle of what was exactly going on there. I mean, does the T cell receptor see the peptide and then an MHC molecule have to sort of come into play to generate things? It seemed, given the, I won't say Avogadro's number, but huge number of conceivable antigenic peptides that T cell receptors are, which we hope that T cell, which there'll be at least one T cell receptor to be able to bind. If the MHC1 molecules are actually binding and presenting the peptide, as we now know is the case, how could because we have mother and dad, how come we have maybe a total of 12 MHC1 and MHC molecules and so forth? How can let's consider just the 6 MHC1 molecules you'll have, the A, B, and C genes from mom, and the A, B, and C genes from dad. Total of 6 genes making hopefully 6 different MHC1 proteins presenting each a foreign peptide. How can a single MHC1 gene product, that protein, how can it has to bind the peptides very tightly, because if they're loosely bound, by the time they get presented out, they'll just diffuse away, and the killer T cells won't be able to see that viral peptide. So how can you get very tight binding and yet very broad accessibility? I told you we now know that the 10,000 different peptides, in particular MHC1, let's say A gene protein, can bind maybe 10,000 peptides very tightly. And then the A2 gene you got from your father, let's say, that's a different set, maybe overlapping, maybe entirely different set of peptides. So we have A, B, and C proteins times 2, because there's mom and dad, so 6 MHC1 proteins, and they can each bind ideally 10,000 peptides, so you could bind 60,000 peptides from the universe of all peptide sequences. So how does so it began to become clear that it probably is what we're saying here, that the MHC1 A gene from mom protein, okay, can bind tightly 10,000 peptides. How did you get both 10,000 peptide binding and get very high affinity constants? And the answer involves the concept of the anchor residue. Okay? The anchor residue here. So let's take a look at what we have here. So here's a picture again of an MHC molecule in white. Okay, the alpha helice mountain range here, and the alpha helice mountain range here, and then the beta sheet floor here, the floor of the valley, and there's the red peptide, amino acid long peptide bound there. So if you look at a particular MHC1, let's say A gene, and this allele number 17, and remember each of these MHC molecules, the gene for a particular A gene that your mom has and the A gene that your dad has are going to be different from each other because there are dozens, hundreds of different allelic variants of that gene. Okay, so this is allele number 17 here. Okay, and here are 3 of the 10,000 peptides that will bind tightly to this particular MHC1 A gene protein, and this allele number 17 version of it. Okay, and well, you can see that the amino terminal end of this amino terminal amino acid here can apparently be anything, and if you look at 10,000, they all have complete randomness here. And what amino acid could be in position number 1. Same thing in position 2 and 3 and 4. But at position 5, it looks like it has to be a benzene type ring on the side chain. It can be tyrosine or phenylalanine. Okay, for both of those amino acids, the side chain is basically a benzene ring. Okay, the benzene ring has a hydroxyl group, but it's tipped if it's tyrosine. It's just plain benzene if it's phenylalanine. Okay, and then anything at this position, but now the carboxy terminal end must be leucine. Okay. Well, these are the anchor residues. So for this MHC1 molecule, we must have tyrosine or phenylalanine at position 5, and we must have leucine at the end. Okay. Now, let's take a look at another MHC protein. Let's see, this is the B gene protein, allele number 37. Okay, out of 200 possible variants or whatever. Now again, the amino terminal end can be anything. The second amino acid end must be tyrosine. Not tyrosine or phenylalanine, gotta be tyrosine. Okay? That anything. But now down here, it's usually there's a little bit more conservation here, but this is an anchor residue. It's basically a sort of a short hydrophobic residue, valine isoleucine, short hydrophobic side chains, basically. So this is the anchor residue. So what's going on here is that I'm going to show a picture here. This in your notes, but so here, this is the cross section. This is the floor of the valley here. Okay, alpha helices in front of it, alpha helices behind it. Here's the peptide sitting on the floor of the valley. Okay, and there's a little pocket along the valley's surface here, along the length of where the peptide is going to bind. The little pocket here is just the right size for, let's say, the side chain of the amino acid lysine. Okay, so this might be lysine. Okay, guess I could write. Yeah, I think I wrote K here. So this could be K. K is the single letter abbreviation for lysine. Lysine is like 3 carbons and then an amino group. Okay? The amino group is positively charged. So there'll be a lysine side chain tube-like structure on the surface, a little rabbit hole on the surface of the valley. And at the bottom of the rabbit hole, there's a bunch of lysines or other positively charged amino acids right here. Well, I bet you I made this one. This must be a spark. Right. Never mind about K. Let's just erase K. And instead, this will be aspartame. There's the A for aspartame, which has a carboxyl group. So this is a negatively charged tip. And it will find at the bottom of the rabbit hole little plus charges from the amino acid side chains that make up the MHC1 protein. Okay? And that's an anchor residue. Okay? And then up here maybe another anchor residue might be a little pocket here, which has negative charges here at the base, and it might be K for lysine, which has a positive charge of ammonia group here at the tip. in the cases where the anchor residue is like phenylalanine, you'll have not a little tube-like rabbit hole, but a little flat-like, slot-like thing here that the benzene ring will just slip into very nicely. Okay? If it likes tyrosine, the bottom of that slot, you'll have another little dip with an hydroxyl group at the end of the benzene where you can so you see there's a lock and key type arrangement between surface features of the MHC1 protein and the side chains of particular amino acids where it's got to be. And this gives you the really high affinity binding. Okay? Now, there is binding. Of course, all peptides have an amino terminal end and a carboxy terminal end. So the amino group is positive, and there'll be a little pocket here which has some negative charges in it. So the amino terminal, the amino group will fit into a little pocket with a negative charge, and the carboxy terminal end has this carboxyl negatively charged carboxyl group, and there'll be a carboxyl group shaped pocket down here, which is positively charged in its space. But if you just had the pockets for the amino terminal end and carboxy terminal end, you'd get pretty good binding, but it wouldn't be the tight binding that you really need. Okay, stay there for days, weeks, whatever. Okay, and it's the anchor residues added to the stuff at the beginning and end that really gives you now that really high affinity, very tight binding that you need to have. And because this can be any one of 20, you have 20 times 20 times 20 times 20 times whatever, 20 and 20 and so forth. Those numbers add up to, in theory, a very large number of different peptides. It's especially large numbers for the class 2 molecules where you can have stuff hanging over the ends and so on. Okay? So, there will be a question on the second exam where I may show you this picture and I'll say, what is the significance of the fact that this peptide has to have tyrosine here or valine or isoleucine here? And you'll tell me, oh, these are anchor residues and they share common properties, and they'll fit into a little slot or pocket on the surface of the MHC1 molecule, and these are the anchor residues. First thing I'll look for is you tell me, these are the anchor residues that this MHC1, for these things to bind to a particular MHC1. Okay? For the B70-37 allele, it's this. For a different MHC1 protein, it'll have different. For this MHC1, these are the anchor residues. For this MHC1, these are the anchor residues, and so forth. And that explains why only 6 different MHC1 proteins can bind in the order of 60,000 overall numbers of peptides or something like that. Any questions? Yes?
So there's no like recombinant DNA when the
No, these MHC1 molecules, building them is not like building an antibody molecule or a T cell receptor. These are genetically inherited, and there is some intracellular mechanism that promotes some increased mutation rate in the MHC1 and 2 genes, so that you generate all these different alleles compared to all of them. Most genes have at least 1 or 2 alleles in the population, but they don't have hundreds of alleles. So we'll talk a little bit about that towards the end. So here are some of the different peptides. This is a class 2 molecule. Remember, the class 2 molecules are called DP, DQ, and DR. So this is now DR. These could be, the class 2 are DP, DQ, and DR. That's the names. Not A, B, and C, but DP, DQ, and DR. Okay, so here's the DR gene product at allele number 3. Okay, there are many alleles of the DR gene. Okay, and remember we said that the class 2 molecules they have they're open-ended. So long peptides. So here's the part that's actually sitting in the valley, shown here in gray, and these are overhang amino acids. Maybe it's all to the, maybe all down the carboxy terminal end here, or mixed, or whatever you can see here. This is a whole bunch of different, 1, 2, 3, 4, 5, 6, 7, 8, 9, I guess 9 different peptides, all of which tightly bind the number 3 allele of the DR gene protein. And here we see the anchor residues here. This was right about here. It has to be aspartate, as you can see, or glutamate. And then here, well, this is not as highly conserved as one might like to illustrate the point, because it can be tyrosine or leucine, or, my God, proline. But anyway, these are hydrophobic at least, or something like that. This just shows it's honest science. It's not as pretty as you might like it for the theory to be. Now then, we talked about this business before. Okay? The peptides that are going to be presented on class 1 molecules come from intracellular proteins. Okay? Remember, I said at all times, in all cells, infected or uninfected, we are more or less grabbing at random proteins, ubiquitinating them, and then the ubiquitinated proteins, they're now pretty much dead man walking. They're only it's only a matter of time before they brush up against the opening of a proteasome structure here, which is a sort of a tube-like structure with a hole in the middle. The peptides, the proteins are going to be unfolded and stuck through the hole. The walls of the proteasome are all different kinds of proteases with different specificities. So as the protein passes through the length of that tube-like opening here, it's sliced and diced by the proteases that make up the wall of the proteasome structure. So whole proteins get unfolded, stuck in, and at the other end emerges short peptides. Now, and then these peptides are transported across the membrane of the rough ER, and so they find their way now in the lumen of the ER. And here's a newly made MHC molecule, MHC1 molecule, and it will not be permitted to leave the lumen of the ER and come up to the surface until it has found a peptide to which it can tightly bind. As soon as the right peptide for this particular MHC1 protein comes along, it'll bind in the floor of the valley, and that will permit this thing to tightly assume its structure, its proper structure, and once it has its proper folded structure, it's permitted then to leave the ER and pass through the Golgi compartment, and then by vesicle trafficking end up on the surface of the cell. Right? And then we said, on the other hand, for class 2 molecules, well, class 2, here's a newly made class 2 molecule. Why don't these peptides also load on to that newly made class 2 molecule? Well, that's because the peptide binding site is covered by the so-called II protein, okay? And it blocks the binding of these peptides, and then the MHC2 molecule with its II chain arrives here. At some point we degrade the II chain, but we leave the little umbrella handle of it in the groove here, and then this is now going, the vesicle containing the newly made class 2, up till now its peptide binding site blocked, will now fuse with the phagosome, and here's extracellular antigen coming in, being degraded with the peptides. There's a process that occurs here where we get rid of the sort of the umbrella handle here that was blocking the peptide binding site. Now we're hoping that among all these hundreds of different peptides from the extracellular antigens. One of them will be able to tightly bind now to the core of the happy valley on the class 2 molecule. And once that happens, a signal is somehow generated that now permits this vesicle with the fourth peptide to come up and fuse with the plasma membrane. And now that class 2 molecule is presenting the extracellular antigen to the immune system. Okay. Now, we're going to go into a little bit of the details here of the class 1s first. We're going to start looking at, okay, how do these peptides get across the membrane here? That was always a problem for this MHC business. Here we have this molecule that's on the outside of the cell, and it's presenting a peptide that came from inside the cell. How did this peptide MHC complex get across the plasma membrane? Well, it's because we use newly made MHC, which is going to be what's topologically the outside world here, or the movement of the ER. And then there's only a question of how does this peptide get across the plasma membrane? And that's provided by a special peptide transporter called the TAP transporter. TAP is transporter associated with antigen processing. And there's two sets in its TAP1 and TAP2. So this is the TAP transporter. It's a peptide transporter. Okay. So it brings, it's one thing to have a sugar transporter, that's a small molecule and so forth, but we've got a transporter that will bind a big peptide and move that across the membrane. Okay? This TAP transporter will bring a wide variety of, it somewhat preferentially prefers peptides that are 8 to 10 amino acids long, kind of thing, so that they'll bind to the class 1 molecules. And for many class 1 molecules, whatever their allelic number, they tend to have either hydrophobic or basic amino acids at the C-terminus. When I use the word residues here, it's kind of a chemistry word, we can just substitute the word amino acid. When I say residue, I mean amino acid. So we had some cellular cytosolic protein here. It gets grabbed more or less apparently at random by the ubiquitin ligase machinery, which attaches first one small ubiquitin protein to it, and then second ubiquitin to the first, and a third ubiquitin to the second. And you get a string of 7 or 8 ubiquitins here, and that's the signal. That the proteasome here has got proteins at the opening of the maw, MAW, the maw of this structure here. They grab these ubiquitins, reel them in, and then unfold the protein and stuff it into the central channel here. And out the other end comes the degraded protein, the peptides derived from that. And then now here is the newly made MHC1 molecule here. Okay? All proteins that are made are designed to be either exported, exocytosed to the outside world, or proteins that are going to become integral membrane proteins. They all are read by ribosomes, and there's what's called a co-translational translocation. As we read the messenger RNA, the protein that's being made elongates through a channel here, and so the protein is coming into the lumen of the ER as it's being synthesized off of the mRNA. Okay? At some point, it releases from the ribosomes and so forth, and then the rest of the protein at that point. So we end up with, in this case, an integral membrane protein. I'm sort of slurring over stuff here. How many of you have taken Bio 3302 molecular cell biology? Please raise your hands. Okay. How many of you maybe didn't take that but had a really good introductory biology course where they talked about the synthesis of secreted and integral membrane proteins involving this co-translational translocation? Raise your hand if you didn't have molecular cell 3302, but at least had a good enough intro bio course that I don't need to explain this process. Raise your hands. Okay. I'm not going to explain it. You don't quite understand it. Anyway, here's this newly made protein. Okay? It's still, when proteins come into the lumen of the ER, there are a bunch of chaperone enzymes that live in the lumen, and they will assist this protein to fold, okay, to its thermodynamically most stable state, quickly and efficiently. One protein here that comes into play for one chaperone here that's involved in all kinds of proteins that will be made like this. It's calnexin. Okay. When a newly made protein is just arrived, it's just beginning to fold, you say it's in the molten globule state, it'll expose hydrophobic site. Amino acids that are hydrophobic will sometimes be on the surface of this only partially folded protein. The accessibility of hydrophobic side chains, lots of them on the surface of a newly made protein, means it ain't folded yet, okay? And so chaperones will recognize these particular side chains. I don't want to get into too much detail here. So chaperone, this is a bit of a chaperone, and it recognizes unfolded proteins that are not completely folded. A newly made protein has to be completely tightly folded in its final thermodynamic stable state before it's able to exit the lumen of the ER. If it cannot properly fold, it never gets to leave the ER and eventually it is degraded. So calnexin comes into play, okay? And we're waiting now for the beta-2 newly made, the mRNA for this protein here, beta-2 microglobulin. Beta-2 microglobulin is a protein which is secreted to the outside world. And so it's also made by membrane ribosomes and finds itself in the lumen of the ER by that same process of co-translational translocation. Co-translational means as you're reading the mRNA into protein, that's translation. As we're reading the mRNA into protein, we're pushing that newly made protein through a little channel in the membrane and it passes through. That's called the translocon. So that's co-translational translocation. And so here's newly made beta-2 microglobulin. When it comes in here, it can now support the platform. I don't know why they're showing, I think they're indicating here that it's in place, but it's not really, we're not still in our final folded shape because we don't have a peptide in the groove here. When finally peptides are coming in from the TAP transporter, finally when they come in, if there's a peptide here that can bind this particular class 1 molecule, here it is, that's bound, okay? And that now permits the whole protein to form its stable final structure. Okay? Now, in addition to first calnexin, which is a membrane protein is stabilizing things, then calreticulin comes into play. Calreticulin is a soluble version of calnexin, basically. And then another protein called tapesin comes into play, where tapesin here, the TAP part of tapesin refers to the TAP peptide transporter. So tapesin is a protein here in green that binds both to the beta-2 microglobulin and to the TAP transporter component here. And now tapesin is holding the newly made class 2 molecule right near the place where all the newly made peptides are coming streaming in. And so it increases the probability that one of the right peptide, when it comes through, will be able to immediately find its binding site on the class 1 protein here. Okay. So now we now have newly made class 1 molecule with its peptide ligand, and it's now fully tightly folded, and it's now permitted to leave by vesicle pathways, leave the ER, and then fuse with the compartments of the Golgi. And then plasma memory. Now, the immune system, back in the 1950s and 60s after World War II, when Stalin was ruling Russia, there was an arms race. The United States had developed an atom bomb which they used to put aid to the war with Japan. That was the end of the Pacific Front. That was the end of World War II. But by espionage, the Russians had gotten the main idea about how to make their own. So they, by the time we had dropped our bombs on Japan, Russia was probably already had their own bomb at that time or shortly thereafter. And we figured by our own espionage, we learned that they had it too. Big bummer for the U.S. military. And so now, who's got the more bombs? Who's got the biggest bomb? It was an arms race. Every time we would come up with a new, better bomb, they would come up with an even better new bomb. So that we would come up with and then finally Reagan said, well, we're going to come up with a defense against the Russian bomb so that we'll be protected. And so that drove the Russians to a frenzy of arms stuff, and that led ultimately to the economic collapse of Russia. So that's what the immune system is like. Our immune system recognizes foreign pathogens and defeats them. So the foreign pathogens, which are like bacteria and viruses, they proliferate much more rapidly than whole animals do. So they're quickly able to get selection of resistant mutants that can defeat the immune system. So that over time, at the cost of the slaughter of hundreds of children and so forth, a mutant child arises which has a mutation in the immune system which can cope with that new twist that the bacteria has evolved and the virus has evolved. And so then the virus comes up with another improvement and so forth. And so it goes over hundreds of thousands and millions of years and so forth. So viruses, for example, here, have evolved mechanisms to defeat. I mean, the whole point of this aspect of the immune system is to detect, for our immune system to recognize that this cell is a virus-infected cell. And the way we came up to do that is we developed these genes that are coded MHC1 molecules. And remember I said MHC1 molecules are at all times presenting peptides derived from cellular proteins. And those peptides are self-peptides, and they are ignored by the immune system. But if you get a virus infection, among all the normal cell peptides that are being presented, now we're also presenting on our class 1 molecules of the infected cell foreign viral peptides. And they are then recognized by killer T cells, and they will then kill this virus-infected cell. So it would be greatly advantageous to viruses to prevent their peptides from being displayed on class 1 molecules. Okay? So viruses do various things to avoid having their peptides being presented. Some viruses have genes that encode proteins that can muck up the proteasome process of cutting peptides. Okay? Other viruses encode genes that produce proteins that will muck up the TAP transporter so that the peptides are being made, but they're not being brought into the room of the ER. Okay? And then the herpes virus can do this business here, messing up with the pathway. Okay? Other viruses mess up with this pathway here, whereby the MHC, the peptide-bound MHC molecule, leaves the ER and moves by vesicle trafficking to here. So the HIV virus does this, and adenovirus also does this. Now, we have evolved countermeasures, but this is one step of the arms race that occurred sometime earlier. So viruses do this, and it buys them a little bit of time, but not enough time, because we have made ourselves have countermeasures to this viral device. We'll talk about later. Okay. So for the last 20 minutes, we've been talking about the right-hand side of this figure here. Now let's talk about the left-hand side here. How class 2 molecules obtain their peptides presented to the outside world. Now, as I told you, all cells in your body do this business here, because all cells in your body can become affected by some virus or another. We're exception. Our cells, like in our body, reticulocytes don't have a nucleus, and therefore they don't have a protein synthesizing machine or any of that kind of stuff. They're just bags of hemoglobin. So they're not a useful target for the virus, because the virus needs to parasitize ribosomal translation of their products and so forth. But a special set of cells, remember the professional antigen-presenting cells, they express, in addition to class 1, these professional cells also make class 2 proteins, okay, which are presenting outside world, like bacterial infections and so forth, or whole virus particles, and then they're being brought in this way. But these are, even before the virus has infected the cell, we can make coat protein peptides can be presented just from virus particles that are floating around. So these professional antigen-presenting cells, their job is, in addition to protecting themselves from being virus infected, their job is to present peptides to the CD4 T cells. So let's take a look here. So here is a newly made class 2 molecule, and the peptides from intracellular proteins. Here are these intracellular protein peptides here. They're present, but they cannot bind to the Pleasant Valley groove of the class 2 peptide platform here, because that's covered up by an invariant chain, II. Like I said, first time I talked about this on the first lectures, I have no idea why invariant chain is abbreviated capital I, little I. Maybe there's a capital I gene that encodes a whole bunch of proteins, and those different proteins are called A, B, C, D, E, F, G, H, I. So maybe this is the I version of big protein capital I. But whatever, the invariant chain is abbreviated I sub I, okay, or Roman numeral 1 sub I. I've never been able to find out what it's called, 1I or II. I'm 99% sure it's II. It's like one of these words you read in a book, but you never hear a conversation. So the first time you say it, you totally bungle it, because there are many funny stories about people who mispronounce a word that they've only known from reading. That just means they're smart, as far as I'm concerned. Okay, so here's the invariant chain, preventing class 2 molecules from binding peptides from the cytoplasm. Okay, so now when the invariant chain binds the class 2 molecule, now the class 2 molecule will fully fold the sort of the handle of the umbrella, kind of the umbrella handle here. When that binds the peptide binding group, it's equivalent to an actual peptide binding, so that the class 2 protein will fold tightly and correctly and be permitted to leave the ER by vesicle trafficking. Okay? And now this will then diffuse around inside the cell. As it's diffusing, these vesicles have proton transporters in them, so they bring protons in from the cytosol, and that means they acidify their internal content. So this becomes low. These are protons. H plus is protons. Protons are acidic. And so now the interior becomes, this is a pH 7, 7.2, whatever. And here, this becomes like a pH 6 or something like that, 6.5. And in this somewhat more acidic environment here, internal proteases not shown. There's a little bit of hand-waving stuff going on in these pictures here. But that leads to the breakdown of the II, the invariant chain. The green invariant chain gets broken down, And all that's left is the handle, which is still sitting in the groove of the class 2 molecule. That handle is now called the clip peptide. So the clip peptide is just the handle part of the invariant chain. And that stays in the peptide binding pool. So no other peptides can get up bind to this yet. And then, in the meantime, these cells, for example, these are macrophages, which are class 2 cells. They're ingesting that whole bacteria, whole virus particles, and they're digesting them, all their proteins into peptides, okay? And this lysosomal type compartment here, the phagosome is called, will fuse with this vesicle that contains the class 2 molecule. And now all this is one big compartment here. And now, up till now, I didn't tell you that there's another protein that's following the class 2 molecule in this pathway. It's a molecule called HLA-DM. In addition to that locus on the chromosome that has all the class 1 genes, A, B, and C, and all the class 2 genes, DP, DQ, DR, there's a whole bunch of other genes there that include the invariant chain and other MHC1 or 2 like molecules. One of these MHC2 like molecules is HLA-DM. Remember, HLA is an alternate way of saying MHC. Okay? So this is MHC-DM. It resembles the structure of an MHC molecule, but its main job is to cause the class 2 molecule to open up, spit out the clip peptide, and now it's free to take up, if it can, one of these peptides from the degradation of an extracellular bacterium or large particle. Okay? If this class 2 molecule is able to bind one of these peptides that are derived from cleaving the bacterial proteins or whatever, it will then fold properly, and it will then be able to proceed on through with its peptide. It is now permitted to go up and fuse with the membrane and release the class 2 molecule with the extracellular peptide displayed on the surface of that professional antigen presenting cell. Your book has a picture here. I'm going to ask you about the test. The MHC2 molecules, which acquired their proper peptide, they now are going to be permitted. Up until now, they've been confined to just diffusing around in the cytoplasm, but having acquired their one of their cognate peptides, they're now going to be permitted to go out to the cell surface in the form of a vesicle like that, presenting it at the surface of the cell. And all this stuff takes place in a complicated organelle that we find only in these professional antigen-presenting cells. It's almost an onion-like skin arrangement of membrane structures here. It's called the M2C compartment here. These are the M2C compartments here. Here's the Golgi compartment. The vesicles containing this are going to end up, this whole pathway I'm describing takes place in these kind of mysterious M2C compartments. And we're not going to worry our pretty little heads about the M2C compartment. I'm not going to ask you any questions about the M2C compartment. Just telling you. Okay. This is one of these afternoons. I'm not going to take a really long break like I did for the first break. We're going to have a short break pretty soon. I'm getting tired of talking and my butt's starting to hurt too. So, here's a picture we've been presenting so far. Here's the T cell receptor. Here's the MHC class 1 molecule. And here's the peptide. So you see the cross-section here. Now, in reality, if this is the sort of the structure of the class 2 molecule, and my other hand is the structure of the TCR, they're not coming in like this. The TCR is angled as it comes in. So as the TCR approaches the peptide along this axis is angled with respect to the long axis of the peptide. And that's what we're seeing right here. So this rectangle is the footprint of the TCR. We're looking at it through the TCR from above. We're looking through the TCR. This is roughly the outline of the T cell receptor. And now here's the class 1 molecule, and here's the peptide. So the class 1 peptide, they're at an angle compared to the T cell receptor. And that's advantageous in terms of making sure that the loops on the alpha chain and the loops on the beta chain are going to maximize their ability to interact with both the peptide and the class 1 protein. So let's elaborate on this a little. Here's one of these tour de force crystallographic papers that was published as we were beginning to understand all this. So here we're seeing the X-ray crystals that have been made of both the T cell receptor and the MHC1 molecule and the foreign peptide. This whole thing to make crystals of this whole complex here. You cut this here, so it's just this domain here. And you cut this, so we just have this domain, this protein, and the T cell receptor. You can get crystals of this, and you get the crystallographic structure of the whole complex here. And from the crystallographic structure, you can really see what's going on here. And so here are the hypervariable loops of the CDR3 from the alpha chain and CDR3 from the beta chain. And then we have the CDR1, CDR2 over here too. Let's look at this from above. Now, this is just for the talk about it. This, I'm not going to ask you any kind of real detailed questions about this, but I might have a question why you'll remember that the T cell receptor comes in at an angle with respect to the long axis of the peptide. So here is the outline of the foreign peptide. This here is the peptide here, lying in the groove, okay? And here is the CDR3 loop of the alpha chain. That makes contact with the peptide. And here's the CDR3 loop of the beta chain, and that also makes contact with the peptide. And the CDR1 and 2 loops of the alpha chain are making contact with the surface of the MHC molecule. And the same thing is true for the CDR1 and 2 loops of the beta chain. So really it's the CDR3 loops here that are recognizing the peptide, and the 1 and 2 loops are involved in binding tightly to the surface of the MHC1 peptide. And a lot of the variability that's going to be found in the TCR loops here is going to be in the CDR3 loops here. That's the part that contains the BDJ junctions with the inserted random nucleotides and so forth. Okay. Here I sit down here. This is what I said. The CDR3 loops of the alpha beta chains bind the peptide, and the CDR1 and 2 loops bind the MHC molecule. And it is the CD3 loops which are the most variable part of the T cell receptor, variable domain. The CDR3 alpha has a VJ joint, and the CDR3 beta has a VDJ joint here, and that's where all, as I just said, that's the most variable part of the variable domain. I don't know. I just realized how close we are. Yeah, I'm going to take a little break anyway. Here, medical relevance here, there are some bacteria, pathogens, parasitic bacteria, that secrete toxic proteins. And they cause a lot of cell death. maybe that means that when the cells die, they release sugars and amino acids and all kinds of goodies that the bacteria will like to eat or something like that. There was a thing that was big in the news, I don't know, 30 years ago, where women, this is delicate here, but women who were using tampons, I'm not quite sure, maybe they were keeping them in too long, I'm not quite sure, but they started, there were instances that kept suddenly cropping up about that time, where they were susceptible to bacterial infections surrounding that inserted object and so forth. And this was called toxic shock syndrome. Okay, this could be lethal on occasion. I'm not quite sure what happened to change why this was happening back then, but whatever. Toxic shock and syndrome, the protein involved here is the toxic shock syndrome toxin 1, okay? And that has something to do with tampon use here. It turns out that this toxin here, this toxin 1, is one example of a number of bacterial proteins that function to intoxicate and cause, make it easier, life easier for the pathogenic bacteria by confusing the immune system. Okay, what they do as a group, they're called super antigens. Okay, and so here is one of these super antigens here. Staphylococcal enterotoxin, SE staphylococcal enterotoxin, known as TSST1. This protein will bind to any TCR and then bind to any MHC molecule with whatever peptide is on it. Most of the time it's going to be normal cell peptides. And by holding these two together, will make the T cell think that it has encountered its cognate antigen, okay? And so it becomes activated. But then, so all your CD4 T cells, whatever their peptide specificity, they become activated, okay? And that gives you like a cytokine storm, and that leads to death, basically. I can't stand it. Let's stop there. Shorter this time. I had a bunch of work to do when I got back around this.
Dr. Bird,
Yeah.
question back here.
Yes. Thank you. What's the question?
So the toxic shock syndrome, does it only activate the CD4 and not the CD8?
Well, I'm not sure whether, I believe yes. I believe these super antigens in general recognize class 2 molecules as opposed to class 1. But there's like a 95% confidence interval there. Let's see, 10 minutes.
We got, what, 3 more pages to do? Yeah. Probably. A lot. Anyway.